teachers. As we all discussed yesterday, that we will see into the molecular biology, molecular basis of inheritance, some topics like, transcription, translation, human genome project, and fingerprinting. Those topics were actually need that we plan to discuss, and also biotechnology. So on that basis, today we will start discussing translation process. You may ask why not transcription. Actually, transcription has been selected by ma'am. Ma'am is going to revise transcription. Already she has chosen the topic. So I thought maybe we'll go with the translation process, teachers. So we'll start our, without wasting much time, we'll start our session for the translation. So since already it is discussed, you would have done some little groundwork on that.
Namaste,
maybe.
sir.
Namaste, ma'am. Namaste, ma'am. I have received that assignment of some of the teachers. It is there in the email. Thank you for timely response. And other teachers also, I'm requesting to send it as soon as possible, teachers. So in the molecular basis of inheritance and the evolution and the principles of inheritance, these three chapters together in 12th standard, it's a very vast and very high weightage. In the board exam, when we are talking about 12th standard, definitely we will be discussing about the marks weightage also, because mark is very important. What marks they are going to get it in the board exam is very important. So we need to discuss on that point of view also. So with translation, we'll see first what is translation, what is the different components are there, what are the mechanisms are involving in that little elaborated manner. It won't be 100% as it is there in the NCERT book. It will be much more in that compared to the NCERT reader. So please kindly put your inputs also wherever and whenever required. Thank you. So what is that idea of this particular translation? What is translation? Transcription is very easy in the sense there is an idea. DNA is having this rule of complementarity that governs that DNA replication. So when one DNA is, yes ma'am.
Central dogma, we call this as transcription.
Yes, central dogma is there, but I am explaining about there is a rule, governance of complementarity. When DNA is made into another DNA, it is definitely possible. When a DNA is converted into an mRNA, then both that is also is possible. Why? Because there is a hydrogen bonds are there. It's very predictable, and there is a complementarity governs there also. So replication and transcription both are having certain rules and certain regulation. Based on that, it is definitely possible. An mRNA sequence is definitely having some matching with the DNA. They have some complementarity between them also. But when it comes to translation, it is completely a hypothetical, and it's completely a different area of molecular biology. Why? Because how come a hydrogen bond having a mixture of this mRNA can get converted into a protein? A protein that is amino acid, a chain of amino acid, how come that polypeptide will be formed? There is no any complementarity between this mRNA and also to this particular protein. Then how come it is possible? This idea did not just start yesterday or this recent years. This idea came into the mind of the great scientist in molecular biology, the one who gave the double helix model to people. Can you please quickly recall, teachers? Scientist, double helix model.
Watson and Crick.
Watson and Crick, yes, ma'am.
Watson and Crick model.
So the Crick himself has decided, or he had an idea. He had a doubt. How come it is possible to have this complementarity? How come an mRNA molecule can code for a protein, a polypeptide chain? It's a very big question mark. So he himself made an hypothesis called as Crick's adapter hypothesis. It's a very important basic prerequisite. A student must be explained about it. In the beginning itself, in the NCERT reader also, we have this. How come the adapter molecule, idea of adapter molecule? The adapter molecule plays a very central role in that translation process. So this idea, how it came into the mind of the scientist, that we need to little highlight and we need to explain to that. So that also has brought into your notice also this hypothesis called as Crick's adopter hypothesis. You can, even if you want, you can just note it down. If you feel it will be needed somewhere in the students, while explaining the students, it may be required. So if you see amino acid teachers, the amino acid has a very wonderful property. It will be able to very well detect its side chains. That's the reason why amino acid is able to form a peptide bond with the other amino acid. So they have both mixture of hydrophobic and hydrophilic. All the molecules will be present in an amino acid. But whereas this nucleic acid is very strange one, which is very a string of nucleotide which has a hydrogen bonding in that. That is the maximum thing it has. So a nucleic acid can recognize another nucleic acid. RNA can recognize another DNA, or a DNA can recognize an RNA, or RNA with each other. RNA can recognize another RNA, and DNA also can recognize another DNA. But at any cost, a nucleic acid cannot recognize an amino acid. The reason is that nucleic acid cannot distinguish the side chains present in the amino acid. The reason is that amino acid, the nucleic acid cannot distinguish the various side chains present in the amino acid. That is the reason why the Crick has started to think that there is somebody is playing a crucial role who is actually having two arms, with one arm having holding that to the nucleotides, with the another arm that person is holding this particular amino acid. So there is a person whom we can call them as an adopter, that adopter molecule, who is having two arms. One arm is used for holding this nucleic acid and reading what is there in the nucleic acid. The other arm is there to hold that amino acid, that forming different amino acid to form a very long polypeptide chain. So this idea is very important. Students must understand how and why that adopter concept has come in existence, that we need to explain to the teachers. Teachers, shall we go to the next one? Are you clear with this?
Sir, once again, can you repeat what this adapter, sir? Once again, can you repeat this adapter?
This idea of the Crick is this adopter molecule playing, otherwise that mRNA cannot be converted into a, mRNA cannot code for a polypeptide, amino acid polypeptide chain. Somebody is there in between mRNA as well as this amino acid, who is playing a crucial role. That person is adopter molecule. That person is in one arm, he is able to read that nucleotide. The other arm is able to handle this amino acid, read the side chain of the amino acid.
Okay.
That person only we call it as adopter molecule. So this is the idea gave that by Crick. This idea is the basis where many recent scientists who is not even having any relevance to biology, they also have played a very important role in discovering of this genetic codons, the tRNA molecule, the role of ribosomes, and all these things later only came in existence. So can anyone teachers say what is this tRNA molecule previously called as? You can put it in the chat box. You can put it in the chat box, teachers. What is the previous name, earlier name of this particular?
Soluble RNA or heterogeneous RNA, sir?
SNRNA. So sRNA is most appropriate, ma'am. sRNA, very nice.
Soluble.
Yes, teachers. So what is that answer for this question? It is actually sRNA. Yes, soluble RNA or sRNA is the name given to this particular adopter molecule.
Heterogeneous RNA is sir.
Is also there, and also we can even call it as
HNRNA.
Yes, very nice. And also we call it as adopter RNA also. Previously we used to call it. So actually this RNA was discovered much before when they discovered this particular concept of translation. But the name was given very later only. Thus they took a lot of time for them to recognize that sRNA as the tRNA. That took really a lot of time. So when we are teaching to the children, we need to give equal weightage to the children about the already, as we always say, the scientists who have discovered, because it is not so easy to put something very the in vitro also later only it was developed. So how was the scientists that have, first of all, whenever they are discovering something, they won't all of a sudden they will not enter into the laboratory and they are not going to discover. In that way, it is not going to work out. Whatever they, it's a philosophical basis only. First of all, they need to give an hypothesis of that. Then various scientists will try to involve in that, try to bring about that experimentation of that, and later only it will be discovered. The students must also value the role played by the scientists. They were all know, they previously know there is a need of 20 amino acid. Even we can ask the children of question, 20 amino acids are there. If there are only 4 codons are there, how come these 4 codons can code for 20 amino acid? Before we even start teaching to them, we can ask them this question. So we can see what way, how come they think about it, and what are all the different types of answer they bring out. That also we can check. When one if it is we go with the concept that the codons are single in nature, only one single codon only codes for amino acid, then hardly there will be 4 amino acids only. Then we can give the idea to the children, what if that codons are doublets, 2 codons are there? We can ask the children to give the answer for it, and also we can make them to write it, what AT, TG, GC or CG, like that. They can write all the different 4 power 2, that is 16 different amino acid. How come it may code? That also they may write it. At last, we may give that if it is 4 power 3, that is 4 into 4 into 4, that is it's a triplet in nature. The codons work in a triplet nature. That's why 64 amino acids are formed. At the time, it was regarded as very bold, because we need only 20 amino acid, but whereas 64 amino acids are formed by this particular this. When this hypothesis was formed, many scientists did not agree with that. We need to tell to the students all this thing. The scientists argued that how come it is possible. They didn't agree with that. This all happened in the history. But later, you can give credit to our Indian scientist also here, the Hargobind Khorana, Marshall, that is Marshall Nirenberg, and Robert V. Hawley. All these 3 scientists together, they were able to discover which one is that, how this codon concept is actually practically how it is working out. What are all the ways they discovered, that also we are going to see now. And can any teacher say in which year they got Nobel Prize for this? Yes, ma'am. They were awarded a Nobel Prize for this.
Yes, sir. Yes, yes.
Can anyone quickly recall, try to recall? Somebody is Googling, I can say that, it's okay. You can say that. By Googling also you can say no issues. 1970s, I'll give the clue.
71, sir. They
Yes,
had
ma'am, sir.
synthesized gene for alanine artificially first.
Yes, ma'am. 71 or 74 or 73 teachers. You can check it.
71,
73, sir.
Okay, ma'am. Thank you.
71 is,
Mhm. Yes.
Not
first
remembering
he had
the
synthesized.
year.
Only in
Yes, yes,
71.
yes, correct.
Agarwal and Corona, both of them, they did this research. And
Yes,
they
ma'am.
found out. Thank
Yes.
you, sir.
Thank you, ma'am. Thank you. So these 3 scientists, they have very important one. Now we need to say about the codon concept. Hereafter, we are going to enter into the very core content teacher. Slowly, history and all, usually sometimes it will be very boring. But here, that is not that much history. We are going to simply touch it. Why? Because students also should value them. Then only all our students are getting toppers, and they are completing PG, everything they complete, but they don't enter into the research field. The research, the scientists and all, very less number of scientists we have. So by encouraging all this way, by motivating, even many scientists will be produced. Who knows? Maybe from our class, one Nobel laureate can be there.
Scientist will be.
Yes, that also is possible. Thank you. Now this person who is not having any relevancy to biology, mostly that person is actually gave the term genetic code. Direct molecular biology, it doesn't have any direct contact. The person who gave the concept of genetic code, George Gamow in 1954, he gave that word, the terminology of genetic code. And he said, this is codon. What is codon? The students will generally have the doubt. How can we also better way we can define? We can compare it with the computer. The computer can, whatever we type, we are typing on the laptop or computer, but the computer understand it's in a different language, in its own language. Same way, whatever the proteins we are having, whatever the enzymes are produced, though it is produced, we call with the different names. There is a language in which the mRNA or the molecular biology understands. A mRNA is called as a codon. So codon is nothing but it's a language. It's a language of the molecular biology, how it is understanding what is present in particular thing according to the different enzymes or different proteins are produced. So it's a language we can compare with it, the student. And very funnily, they named this anticodon. In the tRNA, they call it as anticodon. But scientists previously, even now, many scientists, those our teachers and all, they would say that word NODOC. Can anyone say what is NODOC? NODOC, N-O-D-O-C. Codon is something present in mRNA, and NODOC is something present in the tRNA. Actually, nowadays we call it as anticodon. Even our teachers and all used to say NODOC only. Can any teacher say what is NODOC? Meaning of NODOC? You can try, teachers. We are all going to learn together.
Terminator codon, sir?
Okay. NODOC. Any meaning for that NODOC?
No, sir. No idea.
Yes. Actually, it has no meaning, teachers. It's actually a palindromic sequence. You can see from the end to front, you can read last to first. CODON. That only is written in an opposite direction as NODOC. It has no meaning actually. CODON only. If it is present on the mRNA, we call CODON. If it is present on the tRNA, we call it as NODOC. NODOC is nothing but in the reverse written of codon only is called as NODOC. It has no any separate meaning. It is NODOC. Usually
That is
teachers use
anticodon.
anticodon, but the word NODOC, it is not having any meaning. Codon only is called written in the backward directions. Then next one is Nirenberg, another very important scientist, along with Hargobind Khorana. That Marshal Nirenberg's role is unavoidable. It's very commendable, because of which today we have a lot of clarity in various things that we are doing in the molecular biology, both practically and as well as theoretically. So Nirenberg was the person who actually discovered the role of UUU codon, triple U codon, that codes for phenylalanine. He only has discovered. And Hargobind Khorana also helped in the discovery of various codons like this valine codons, that and all he tried to discover. And also he is the first person, without him today even PCR is not possible. He was the first person who actually chemical method of synthesizing the copolymer and homopolymer and all, Hargobind Khorana only has discovered. For that, the credit definitely goes to him. Teachers, history, little history only, not too much. Kindly bear with a little history in between, because that is having relevancy with our topic. And how did Hargobind Khorana has actually discovered this? If you see, it's a very, this also we need to highlight to the student. How it is possible for him to say that what is this code for, what is this particular codon is coding for? How come we say? We cannot see that enzymes regularly, we cannot see that letters written there, we cannot see how it is experimentally proved. If we see, he has taken homopolymers and in a different combination he has taken that. Say for example, uracil, cytosine and uracil. Then uracil, cytosine and uracil, like that in a different combination. It won't be in a proper order, in a different order he has taken. And each and every single codon, how it is, what it is going to secrete, what it is going to express in the amino acid form that he has actually again and again repeated n number of times. Because of this only he came to an understanding that there are some codons which do in combination of UAG, UAA and UGA. In this combination, whenever he is trying to make the express in the laboratory condition, amino acids were not formed. By this, he gave an idea. There is possibility there are three codons do not code for anything, but he didn't call it as a stop codon. Later, another scientist, there are three very important scientists, very importantly, Richard Epstein, Charles Steinberg, and their friend Harris Beristain. These three scientists, triple people actually, they only gave the name of this stop codon. You may remember, I believe so. Do you remember, teachers, that Amber, Ocker and Opel?
UAA, UGA, and
Yeah, it was a old name, actual name. Even nowadays, if you go to the German, Europe and all, I hear like they will say this words only. They will usually use to say that different stop codons. So these three scientists are regarded who actually experimentally proved what are the stop codons are there, and they gave the names based on the colors. And actually, it is the name of their surname also. And they gave the based on the color, they gave the names, like three names are very importantly given. For UAG, they gave the Amber, and for UAA, they called Ocker, and for UGA, they called Opel or Umber. So these are the common names given to these things. You may note it down. When you say this thing to children, it will be really somewhat interesting. When really they heard about the names of the different people with the Amber and all is there. Actually, that is the name given to this. The students will feel little interesting in that class about knowing different name, because they are all expecting beyond textbook from us. I believe so. And this termination codon as stop codon, we said, it's previously they were also called as nonsense codons. Now only it is called as a stop codon. Previously, they also were called as nonsense codons. In a NEET, this kind of questions are generally expected. So please note it down. So nonsense codon is actually nothing but the same stop codon only is called. Sometimes we will misunderstand. Nonsense codon will be anticodon. So you should not, we should not get confused there. Nonsense codons are nothing but this termination or stop codons only is called as nonsense codons. Now, this termination is alone. It's not alone. This fact, this particular codons alone is not having role. Along with that, there are some factors, protein, the factors which are made up of protein, which also plays a very important role in the termination of translation. Now itself, these things I'm giving here, anyway, we are going to say elaborated manner later, but you need to keep in mind, that's why here itself we are just saying that we call it as release factor, RF, release factor. There are two release factors present in prokaryote. The reason for two release factor is they have this kind of operator concept. So there is always a need of two release factor. Whereas in the eukaryotes, we have only single release factor, because mostly our site polypeptide or peptides are continuous in nature. So we have only one single. The eukaryotic organism have only single release factor, whereas the prokaryotic organisms have two release factors. Now you can, teachers can say the answer for this question. You can put it in the chat box also. Yes. What is the answer for this question? Easy, isn't it, teachers? This is only to test your presence in the class. That's all. Nothing. Definitely, you know the answers. One answer only I got, teachers. Only one ma'am has replied. Jansi ma'am, thank you. Others did not reply. I'm waiting for the reply from all teachers. You can even orally say, no issues. Yes, teachers.
Marshall Nirenberg, sir, the same person.
The scientist who coined the term genetic code. Yes, ma'am.
Coined.
Yes.
Marshall Nirenberg, sir.
Recently, we saw in the
He
previous
disco-
slides also. Yes, yes, ma'am.
Nirenberg.
Okay, I am in the same
Marshall
page where
Nirenberg.
the answer is there.
He coined Gamow, George Gamow.
Yes, ma'am. Yes. Thank you. It only to simply
Yes,
test
Marshall
whether
Nirenberg, sir.
yes,
Marshall
actually
Nirenberg, he has discovered,
Nirenberg.
but coined.
yes, discovered that it is also proved by him how many codons, how they are functioning. That the word terminology alone is given by George
Gamow.
Gamow only. Thank you, teachers. So next, now little two things. This students will have the doubt. Definitely, they are asking, how come it is possible for the scientist? Even now, they may not ask the doubt. But when you are explaining about the later part of Marshall Nirenberg, after that there is a enzyme name. You may remember very well that one particular enzyme. Can anyone recall the name of enzyme, teacher, which plays very important role in
Sir, I didn't get you. Can you repeat once again?
enzyme which plays a very important role. Because of this only it was possible in vitro discovery of all this thing.
Sir, restriction endonuclease, sir.
Okay. Okay, I will put it in this way. This enzyme will make that translation in a template independent manner. There is no template is not required at all. Whatever it may be in the template, without using the template, it may form that polypeptide.
That peptide transferase.
Okay. Yes, ma'am. There is a name, very particular name for that. You can try it, teachers. It's a name by the some scientist. Some oil, bike oil also comes in that name of that particular the part. Okay, the enzyme, it's two words starting with S. First word start with S, second word start with O. Yes,
Is it
okay.
enzyme, sir? Is it
Yes.
enzyme, sir?
Yes, ma'am, it's enzyme only, definitely.
Then second letter, sir.
O.
Second letter.
Second word, first
Second.
letter O.
No, first word, second letter, sir.
First word, yes, E. First word, second letter.
Irina, sir.
Okay, now we will be seeing that. Next slide, we are going to say that. Now, first one, these two things, the chemical synthesis, how it is possible, students will be definitely asking, how come it is possible that we make that in the laboratory? Means it is also the discovery of oligonucleotide. This, when you are talking about Hargobind Khorana, we need to highlight what is his role. If his role is not there, definitely today molecular biology, 90% of the work what we are doing today will not be possible. So his role is that oligonucleotide that he has actually made homopolymer and copolymer. These things can be made in the chemical synthesis method, that is in the laboratory. Using this, even today what we are doing PCR, whatever that molecular diagnosis we are doing, for everything the basis came from this particular discovery only. So we need to definitely highlight the role of this oligonucleotide to this children. Now, how come this methionine AUG is the start codon? We already know it also codes for this particular amino acid methionine AUG. This is the code. Now, this two enzyme I said, do you remember one enzyme name I said? Can you find that name now?
Servo.
Servo.
Servo Oka enzyme. Yes.
Oka.
Servo
Servo
Oka
Oka,
enzyme.
yeah.
Yes. This enzyme, if it is what was not discovered, the cell, the laboratory discovery, laboratory how we are making that polypeptide, that wouldn't have been possible. And Marshall Nirenberg gave the cell-free system of protein synthesis, in which the protein synthesis itself, the translation, entire translation itself will not require a cellular apparatus. It will be taken out of the cell, mRNA, tRNA, everything will be taken out of the cell. Similar stimulated atmosphere will be given in the laboratory. where this protein synthesis will take place. This is one very important discovery. Second thing is Servo-Oka enzyme. In this enzyme, actually, no need of template. If any protein has to be synthesized, a mRNA template has to be given. If mRNA, if it is given only according to that particular polypeptide will be produced. But this particular enzyme, there is no need of any template reading for this mRNA. Without that also, this enzyme will be able to make it. The RNA or also in the protein synthesis, it plays a very important role. So these 2 points are a very important discovery behind the laboratory scale studies or experimentation of molecular biology, whatever the concepts are there related to molecular biology. Photo depicting Marshall Nirenberg cell-free system, how he was doing that. In the another image where the diagram cartoon is written, this is how the cell-free system is made. Cell-free system of protein synthesis, in which from the cell all the required things like all the materials required for protein synthesis will be isolated. Later in the stimulated environment in the vial itself, it will be making the protein. So this is the cell-free mechanism of protein synthesis. Children can be even informed about. These are all the reason why that codons were able to discover. Otherwise, how come the codon be discovered? What is a codon? How this codon is coding for that particular amino acid? These things we are able to discover only because of this method developed by Marshall Nirenberg. Yes, teacher, somebody saying something? Okay, thank you. Okay, next, now we already studied about all this thing. Now adapter molecule we are going to see. Shall we go to the next slide, teachers? Are you ready? Or need some questions to boost you up and motivate you? Do you need some questions in between? Yes.
Yes,
Okay,
sir.
we can say where does this translation process take place in eukaryotic cell?
In cytoplasm.
Cytoplasm, sir.
Okay, sir. As you all know, there are 2 ribosomes are there, teachers. One is a free ribosomes, another one is the bound ribosomes, which is bound into the endomembrane system. In which of the ribosomes will this translation take place? One is a free ribosome that is dispersed in the cytoplasm. The other one is membrane bound. The membrane sits. Yes, ma'am.
Free rib.
In RER.
Yes, RER it is attached. Which one in the RER? It's a RER only. It's a membrane bound ribosome. In that only it is
In
happening.
cytoplasm only, free ribosome only it will take.
No, free ribosomes will be playing a role in a small part only. Mainly it takes place in the bound ribosomes only. The ribosome which is bound to the endoplasmic reticulum, in that only it takes place. Okay. Last question, teachers, to check whether you're all active. Which organelle helps in the formation of ribosomes? Which cell organelle helps in the formation?
Golgi body, sir. Golgi body.
Endoplasmic reticulum.
It is there inside the nucleus.
Nucleolus, sir.
Yes,
Nucleolus.
yes, sir.
Nucleolus. Nucleolus.
Yes, yes. Thank you. Now, this is the structure of a tRNA or adapter molecule, or previously called as sRNA, soluble RNA, all meaning to the same structure only. What is there you are seeing on the screen. Actually, in a 3D image, it will look like a clover leaf model or inverted L model. So in this coiled structure, if we see, there are different arms are present. In this, mainly we need to see the polarity is very important. The 3' and 5' end is very, very important. Students are always having doubts in this. N number of time also we say they get confusion. Even in the transcription, it happens. They will have the doubt in the polarity of that particular one. So 3' end is the place where always amino acid will be present. That student must be very, very clear. 3' end is the place where always amino acid will bind. That is the amino acid acceptor end. And this opposite to that, on the same structure only, but not in the 5' end, on the entire in the middle of it, we have the anticodon loop. The anticodon will have the nucleotides which is opposite to or complementary to the codon which is present in the mRNA. So mRNA will always the translation will occur from the 5' to 3' direction only. Always it has to be highlighted to the children. 5' to 3' is the direction in which this particular translation process is going to take place. And here, though it is not given in the NCERT reader, we need to little elaborate the structure of particular tRNA to the children. Otherwise, the students will always will not able to understand, comprehend how this actually translation process is taking place. So there is a need for explaining them by giving the example of AUG, the start codon. What will be the nucleotides present in the anticodon loop? The anticodon, what are the nucleotides will be there? That we need to highlight to the children by giving an example of AUG. Let it be start codon. Anyway, it is going to start with that, and methionine going to be the first amino acid. So by giving this particular example, we need to tell them how these anticodons are present in the tRNA molecule. And the tRNA 5', 3', and this confusion students will not should not have. The 3' end is the place where amino acid acceptor end is there or amino acid attachment site is there. The student must be very clear. Even in the CBSE, however the question paper is given, they may give the 3' end in any side. They won't give the same diagram what is there in the reader. They may even give the inverted image also. Still, the students must be able to find it correctly. And what happens to the another many students are thinking, they are not able to think that DNA, RNA as a molecule. They simply keep the letters alone in their mind, and they assume that the letters are the true. It's not like everyone know that letters are not going to be there in the molecule, but they fail to understand how exactly the molecules will be looking in the living system inside our body system or bacteria, wherever that, how this molecules will be looking, that we need to explain to the children. Can you all see it, teachers, on the screen?
Yes, sir. Yes, sir.
Yes, you can easily download, teachers, from the internet. There is a NCBI. There is a website called as NCBI, or there is another software called as Rasmol. Like many websites are there, they are giving the 3D image of number of protein. Insulin, you want to say, insulin structure is there. You want to say about any particular type of protein, hemoglobin, there is a structure, 3D structure. How exactly they will be looking in the living system, or how exactly they are going to be there in the reading, the cartoon image. They need to be they need to understand that. So what you are seeing on the screen is actually the 3-dimensional image of that particular tRNA, pictures. So this definition, what we are seeing on the current screen now, students must be very clear about this definition. What is a gene? What is a genetic code? What is a codon? And what is a polypeptide? When they know this, all these 4 things, learning the translation process will become more easy and much easier for us also. And whenever the exam, we of course there is something on our we have to behind that marks. We need to bring the best marks in the children. So when this is clear, children will definitely get the better scores in their exam. When they know all these things, the translation will be very easy for them. So gene, DNA sequence, when we say the word gene, immediately student must be relating it to the DNA only. How that particular segment of DNA, which we call it as a cistron of a expressing sequence, we call cistron, they must be able to relate it, teachers. Same way when we call genetic code, they must be able to relate the genetic code to the language of mRNA. The mRNA, how that understands everything, so that it can make the protein synthesis, it can involve in protein synthesis. That is genetic code. The student must be able to understand that. And codon, another word, that 3 base pair. When we say the word codon, student immediately must recall that it is a 3 nucleotides bases, or even nitrogenous base, they can say. The 3 numbers should be already fixed. Triplet, it's a triplet that should be fixed in the mind of the children. Codon. Then polypeptide, they are all very know already in 11th standard. They are studying that polypeptide chains, protein synthesis, all they are studying. definitely they will know that what is polypeptide. That is, there is no doubt about that. But here we need to again explain them whether it's a homopolymer or whether it is a heteropolymer. Again, we need to make them understand. So this 20 essential amino acids, that is very clear. These 3 bases, the codons, that what is genetic code, these are all the things we know already, but we need to, I'm just trying to put in front of you that student must be very clear about it. When they are clear with all these things, there won't be any problem in learning whatever the next upcoming translation, even up to this human genome project. They won't have any confusion. With a very easy understanding will be there in the students. And next, when the teachers, when we open the book, next terrible thing comes in front of us is the codon chart. How are we going to make the children to understand it? How are the things?
See,
Please,
sir,
teachers.
first position, second position, third position.
Yes,
Then
yes, teachers.
I
That is
made
very
all
easy.
my children to do, sir. I made all my class children.
Very
to do,
nice,
sir.
ma'am. Very happy to hear that. Very happy. That is a challenge in front of us. We need to make the children to understand it, how the codons are formed. One thing. Second thing, what the codons are coding for. But when the codons are coding for, even in the medical, there is no compulsory that the students have to memorize the name of all the enzyme except some of the things. Some of the things are very, very important, but not all the things. But when it comes to codons, they have to keep the codons in their mind very clearly. As ma'am said, I used to give this one, the slide you are seeing now, you may be seeing now. I always give this to children. Even children, some children, they more enthusiastic children, they make a rotating disc, ma'am. They make with that and they try to find which are all the enzymes are there. Even I have it in my laboratory, I have kept it. They will be able to find how these codons and what are the codons coding for a different amino acid. They will be able to keep in mind. And I will be telling you a mnemonics. How come that stop codon alone can be kept in mind? Based on that, you also
Sir,
can
how you
make.
will match, sir?
Now
How you
I'm going to
will
say.
arrange, sir?
What, ma'am?
Okay.
Excuse me, ma'am.
How
Yes,
you
ma'am.
will make the 60 amino acids, sir?
No, 60 amino acid. In this, they will be writing only a selected one. They will be making a rotating disc, ma'am. In that,
Oh,
yes,
okay.
in the 3 things only, the same thing what is there in the book only, the first base, second base, third base, the same thing they will make as a rotating disc, and they will write the name, and the last disc they will make as a amino acid that they themselves will make it. It is not like something we will only tell them to make a chart. They themselves will make all these things for their easy understanding in the keeping in mind.
Yes.
One mnemonics I'm going to share with all the teachers, easy mnemonics only. Now later I'm going to just share it with you. Now there are some now this is the mnemonics which I wanted to share with you. The stop codon, whether other things are important or not, stop codon sometimes always confusing. Students will be keep on asking. They need to keep in mind definitely. That too, last exam we had a MCQ. In MCQ, what will happen? All the 4 options are there. It will be confusing, misleading. So for the children to keep in mind, you can give this particular kind of mnemonics. You are annoying. So UAA. You go away. UGA. You are gone. UAG. A simple mnemonics we can use it in the class, so that children at least the stop codon they will be able to keep in mind. Stop codon they will not forget. UAA, UGA, UAG. This simple kind of mnemonics they can keep in mind. What are all the things coding for stop codon? They can keep in mind you are annoying. So UAA, you go away, UGA, you are gone. So UAG, like that you can keep. Yes, teachers, will you? Is it possible to follow it for you? Will it be useful to you in your class?
Yes,
Yes,
sir.
sir. Yes, sir.
Yes.
Yes, sir.
Yes, kindly please make use of it. And even more, if I find, I will that also will share it with you. This sometime it will be involving. We cannot make a sentence for kingdom plant and all. There we use that word. Keep plate clean or family gets sick.
Yes.
That is very easy to keep in mind. Here usually we cannot make such a big sentences. but we'll try together. We'll try some other possibility how we can do. So next, the translation. Now we are going to enter into translation, how the process of translation is going to take place. And one small questions to you. You can say that answer. Which of the following is not necessary for translation?
Ligase.
Ligase, sir.
Very
Ligase.
nice. Yes, exactly, teachers. Yes, that is not required. Now, next one is the properties of genetic code. Of course, in the book it is given, but not in an elaborated manner. And there are some exceptions also there. Both the things we need to tell to the children, so that children will have the maximum satisfaction and maximum understanding of that particular topic. First one is, first property is, it is triplet in nature. It is always having three letters together, three nucleotide together will join together to form a one codon. So it is always universal. It's a triplet in nature. Then degenerate. It's already there in the book. We always say that. Can anyone add on to that? What is degenerate, teachers?
When one amino
Coded
acid
by more than one
Coded by
codon.
more than one
Since they
codon.
are degenerate.
Very nice. Yes. Then universal. That is almost whether you take it a bacteria or whether you take a human or any amino acid, it's going to be the same. Non-overlapping. Very important. Children need to understand what do you mean by overlapping and what is non-overlapping. Of course, we are explaining. Say, for example, we have A, U, G. And next we are having C, U, A. So in a non-overlapping means AUG will be read as one codon and next CUA will be read as another codon. If it is a overlapping, what will happen? UGC will be read as one codon. GCU will be read as one codon. Then CUA will be read as one codon. Like that, it will be overlapping. Is it possible to have overlapping codon, teachers? Is it possible in living system? Do we have any such example?
Mutation, sir.
Mutation happens, yes, ma'am. Any examples can we give where it is actually happening? Yes, it is happening. Now I'm going to share the example with you. Yes. Next, Camales. They don't have that, that they are continuous in fashion. All the codons will be read in a continuous fashion. Non-ambiguous. It's a very important thing that one particular amino acid will code for only that particular, the codon will code for only that particular amino acid. No two different amino acids will be coded by the same codon. But what about GGA, teachers? Do you have any idea what GGA codes for?
Sir, is it
I believe it is an ambiguous one.
starts with T. Is it tyrosine or tryptophan?
It goes with, I think, glutamic acid. It will be not glutamic acid.
No, glutamic acid is coded by
Glycine and glutamic acid.
No, sir. Glutamic acid is coded by two code.
G.
I think
We'll check it in the table, ma'am. What is that? GGA, right, ma'am? In the table itself, we have
Glycine,
the
sir.
table.
GGA glycine.
GGA codes for glycine. Here also we can have GGA. Yes, GGA codes for glycine. Can it code for any other amino acid? Because in some resources they are saying the GGA as a very ambiguous one. It codes for even one more amino acid like glutamic acid, glycine, both it is coding in a different living system, not in human, but in other living system, it is ambiguous, they are saying. So as of now, we'll go with that. It is only glycine is coded, but if it is ambiguous, that I will let you know if any other research is there. Then gene polypeptide parity. Gene polypeptide parity, a very important one. What is nothing but what is there in the gene, according to that polypeptide should be formed. What is there in the gene, according to that polypeptide has to be formed. This concept is known as gene polypeptide parity. It should not be a counter opposite to that. What is the gene present? The gene what it's code for, the cistron, what it codes for. According to that, polypeptide should be produced. This concept we call it as gene polypeptide parity. Last one is the collinearity is also another very easy one only. If we take a DNA, it is all in a continuous fashion will be there. Whatever is there, that will be complementary to the mRNA. What is the code present in DNA? According to that, complementarily, it will be there in the mRNA. What is there in the mRNA? According to that, amino acid sequence will be there. What is there in the DNA? According to that, mRNA will be there. What is there in the mRNA? According to that, polypeptide, amino acid polypeptide will be there. If any changes happens in the DNA there, a small changes happens, that will affect this amino acid produced. Whatever the changes happens in the DNA, that will affect the mRNA sequence, and that will affect that mRNA sequence will affect that amino acid sequence. This concept is called as collinearity, that what is there in the DNA according to that mRNA will be there. What is there in the mRNA according to that amino acid sequence will be there. This concept we call it as collinearity. But there are some exemption, as already I said. In the case of first case, if you take that very famous organism, that paramecium, in which you will find this UAA and UGA, which is actually what is the teachers? What is the job of this in the human, the other living system?
Stop codon.
Stop codon. That stop codon is actually going to code for the glutamine in the paramecium. Same way, if you take the pi 174, now I was saying I will give some example of the virus SV40, this virus if you take, they have this overlapping gene. Nowadays, it's a very important field of study in virology, where people are finding that overlapping genes. Because the virus is so small, they have a, the reason is the accommodative. They don't have too much of sequences, which whatever the sequences they are having, they are using it in an overlapping way to code for a variety of amino acids. So it's a very important branch of field of study nowadays going on. Overlapping genes, they are mainly present in the some of the viruses as stated. No need to go to outside example. Even within our living system, we have our mitochondria. The mitochondria will have self-replicating. There also this AGG and AGA codon, which is actually code for arginine in our general system, but that is acting as a stop codon in our own system itself, the mitochondria. That is actually doing. Then in the yeast and all, if we take that mitochondria, the UGA actually has to code for the tryptophan, but it also can act as a termination codon in other things. In other, it will be adding as a termination codon, which is actually acting as a, which is coding for the tryptophan in the mitochondria of the yeast. So mitochondria, it's always, we know very well that it's not actually a part of a living system. It was previously a separate prokaryotic organism, which was acting as a, it was a parasite. It was having some association with the thing, and later it has become a part of our cells. Previously, it was a separate organism. That is a theory. It is still been proved. Because of all this exception, it is proved. So first one is in the paramecium, the generally which is stop codon called as in other organism, which is actually coding for glutamine. Whereas in the some virus like pi 174 and SV40 and all, there are overlapping codons present. Actually, as per the rules, there should not be any overlapping codons, but it is there. In human mitochondria, this AGA and AGG, which is actually coding for arginine, which also act as a stop codon. In the yeast mitochondria, the UGA, which is actually a stop codon, which also codes for tryptophan in this yeast mitochondria. So these are all the some exemption which we can tell to children so that they will have an idea about what it is. Not in a living system, nothing is so perfect. So now one questions to you. What is the verbal position? Can anyone say? Teachers, you can try.
Jumping position, sir.
Very nice.
Sir,
Yes.
you told one tRNA it can able to recognize like more than one codon.
Very nice. Yes. Yes. Yes, teacher. Actually,
That
Obel
is the
position.
reason you told like something one amino acid is was recognized by one more codon
Okay, very nice.
because of that.
Actually, in the codon, we said 3 nucleotide sequences very important, which determines actually the type
Even
of amino acid it is going to actually code. Yes, yes, ma'am.
Third nucleotide is very important
Nucleotide,
in that.
if it is going to get changed, I
Yes,
think case of
yes.
degenerative code will not have problem.
Third
Yes, very nice, ma'am. Exactly, that is what I was expecting. Very nice. The first 2 is that one which is actually going to code or decide what amino acid is there, and third one is actually it has no mostly there is no role played by that to determine the type of amino acid is going to be played. That's why we call it as a Obel position. It is also now very currently it's a research area where more and more research are being conducted in that. Student must be definitely aware of this, because by the time they come to the college and all, there will be definitely they will learn all these things. There will be a gap between what they have learned in school and what they are learning in the college level. So little idea only we need to give. No need to elaborate completely. Even in a NEET and all, it is definitely there. But little we need to give a little elementary idea. We need to give to the children what is the Obel position can be. So next, the Obel position. If you want, you can even note it down. If you feel you want to take some information about it, it's up to you. It's not compulsory. If you feel, you can just note it. This is not applicable to mainly the tryptophan and the methionine, because these amino acids are coded by only one codon. So please note it down. This Obel position applicable only when more than 2 codons codes for the amino acid, the same amino one type of amino acid. In that case only the Obel position is applicable. When the tryptophan and methionine, anyway we cannot apply it because only they are all single. Only one AUG, only one methionine will be produced. That's all. Methionine will not be coded by any other amino acid. Same way the tryptophan is going to be coded by that UGG. That's all. There is no any other codon is going to code that. Whereas the other amino acid, which can be coded by more than 2 codons, same amino acid coded by more than 2 codons, in that case the Obel position plays a very important role in the determination of that amino acid, where we can say degenerative. Generally they are not, but here we can say degenerate.
Sir, excuse me. That is
Yes,
why
ma'am.
I said when he asked for glutamic acid, non-ambiguous amino acid is only this. I didn't remember whether it is tyrosine or tryptophan. So I
Yes,
think
yes.
non-ambiguous is coming for only this.
Yes, yes, ma'am. Yes,
One
ma'am.
code for one amino acid.
Yes,
I
ma'am.
think your answer like glutamic acid will not suit here because glutamic acid is having 2 code.
Mm. No,
Am I
glycine,
right, sir?
mainly the glycine, there is a different argument is there. It is not like in the textbook reader. There is a different arguments. Can that GAA be, GGA be ambiguous or not? In the different living system also, they are trying to find that answer. It's an ongoing research only. That's why I put a question mark.
Okay, sir.
Okay. So can anyone say the answer? Very easy answer, teachers, quickly.
AUG.
Yes, option D, AUG. Very good. Thank you. So in between to check whether the history has taken you to the dream world or what, to check that only in between with some questions. This question, very easy again. Yes.
Not
Kamal
overlapping.
is
Sir.
Overlapping.
Overlapping.
Yes.
Overlapping is incorrect.
Overlapping.
Yes, its answer is B, overlapping.
Overlapping.
Yes, it's incorrect. The question is about incorrectness. So all the other are correct. Universal, Kamales, triplet, only overlapping is the wrong answer. That's why. Okay, teacher. Now we are going to enter into the process, how it is going to take place when we are explaining to the children. We need to explain in the 3 steps, mainly initiation, elongation, and termination. In the 3 steps, we need to explain it to the children. Initiation, elongation, and termination. Here, 3 things are very, very importantly we need to understand. Initiation also, it is not like simply that mRNA along with amino acid, after the charge amino acylation, it binds in the A site. Then it's not like just only that. there are various factors are also involving in that. Those factors, enzymes also, we need to little highlight to the children, because students will always come with the doubts. Wow, how it is happening? What is that possibility? How the polypeptide chain is, it is catalyzed by an enzymatic only. Everything is happening, the reaction happening in the living system, as we all know, it's a enzymatic only. So here, what is there already in the book, that already you know what is there to this. But little more level, we will try to go as depth as possible to understand much more concepts related to that. So translation, what is, if the children are asking, very easily they can divide, answer that. It is a process of polymerization of amino acid to form a polypeptide. A simple definition they will be able to define very well. If the same thing has to be explained or elaborated to them in a detailed manner, then we can say that the genetic message brought by the mRNA from the DNA, the genetic message brought by mRNA from the DNA is changed into a polypeptide chain with a defined sequence of amino acid. With a defined sequence of amino acid. This we can elaborate to the children. The genetic message brought by the mRNA from DNA is changed into polypeptide chain with a defined sequence of amino acid is called as translation. The children will have more clarity in the second definition. First definition, it is just a superficial and a mediocre one, in which only the polymerization of amino acid to form a polypeptide, that protein molecule is there. But they need to understand it is all based on the DNA only. What is there in the DNA, that only converted into mRNA. Then from mRNA basis, the amino acids are formed. That clarity student must be having it. It will definitely help them in whether they are preparing for NEET or even in the board exam. Definitely it will be very much useful to them. Then what are the machineries are required for this particular translation also? Student must be clear. Not only just mRNA, along with that amino acids, the ribosomes, tRNA, the amino acid tRNA synthetase enzyme. Very importantly, some other factors which are initiation factor, elongation factor, termination factor. All the three factors are there. Initiation also there are some factors, elongation also there are some factors, and termination also there are some factors. All these factors along with the ribosomes, amino acids, tRNA, amino acid tRNA synthetase, and mRNA is very important for the translation process. The first step, it's not initiation, we have not entered into the initiation yet. Before even initiation, there are something is going to happen, that is activation of amino acid. Already amino acids are present. These amino acids are going to bound to the ATP and the enzyme with the help of, so these amino acids along with the ATP, especially in the presence of an enzyme amino acid or tRNA synthetase, this enzyme will make a complex of this amino acid enzyme complex. This process is called as activation of tRNA. Now amino acid will be activated when this amino acid, ATP, and the enzyme, very importantly amino acid tRNA synthesis enzyme, all these things will be joined together, which will activate the amino acid. After activating, it will release the pyrophosphate. Usually PPP will be 3 in number. No, 2P only will be there. That pyrophosphate will be released after this. There are specific, students will should definitely understand. For activation, not just one enzyme is there. There are many different enzymes are there to activate different type of amino acid. But one exception is there, that the isoleucine tRNA synthetase enzyme will activate valine. It's a very important in NEET also. Please kindly note it down. Isoleucine tRNA synthetase will also activate the valine. Both will be done by the same enzyme. Otherwise, all the other have different enzyme for the activation. So this diagram we can draw in the board to make the children to understand how the amino acid is activated. So this is amino acid. So this is that ATP, and in the presence of amino acid tRNA synthetase, The students will always think that there is only one tRNA synthetase. We need to emphasize that it is not so. For each different amino acid, there is a different tRNA synthetase is there that student must definitely understand. So it will lead to the formation of the complex, the amino acid enzyme complex, plus separately it will release that ADP. We generally call it as ADP. So we need to say that the pyrophosphate also is released. So this highlighting this equation, student must definitely know very well. When they are understanding this, we always have the doubt whether children have understood or not. When this and all is explained to the children, definitely we need not to have the doubt. Students will 100% will have the clarity on what is being taught to them. Shall we go to the next slide, teachers?
Yes, sir.
History is over, I think. All the teachers can come back to the class.
Yes, sir.
No more history. Now charging. Next, what is going to happen? Amino acid is ready. Now tRNA must be ready. Amino acid one side getting ready. Now tRNA we are going to keep it ready. tRNA also has to be activated. Why the tRNA has to be activated is because then only tRNA in one arm it will be reading the mRNA sequence. The other arm it will be holding the amino acid. Later all the amino acid will join together to form a polypeptide. So to happen that we need to charge the tRNA. It's all energy depending process only. GDP, ATP, all this thing involving because of that only that process happens. So energy rich, energy depending process only. Now what is going to happen? This amino acid, the already activated amino acid, is going to attach to the 3' end of the tRNA. It is going to attach to the 3' end of the tRNA. Usually that will be a carboxyl end only, and it will form a complex called as amino acyl tRNA complex. Amino acyl tRNA complex, which we generally colloquially we call it as charged tRNA or charging of tRNA, which we call it as, is actually nothing but this amino acyl tRNA complex. The entire thing only we call it as charging of tRNA. So previous step we saw amino acid is activated. Now that amino acid is going to bind with the tRNA and this is activated. So this process we commonly call it as charging of tRNA, but technically we must use the word the amino acylation of tRNA. This is called as amino acylation of tRNA. So here also what will happen after that, the enzyme which was bound to the tRNA will be released. So we call it as the amino acyl synthetase release and AMP released, we call it as. Now first activation charging is over. Now next it is going to be initiation of translation. So already we said tRNA is important. We said amino acid is important. We said that mRNA is important, and very importantly the ribosomes are also very important features. As we all know, the students must understand in this way how different component all they work together. Unity is the strength. How all the people together, different people join together to make the thing success. So same way we need to give a value to the children. Everybody, not one single hand cannot make sound. We all have to join together to attain success. Yes, same way. Now next one is the ribosomes. All these stories we can recall quickly to the children, that 70 years ribosomes, 80 years this ribosome which they have studied in the 11th standard. We can quickly recall it to them so that they will have a clarity on that. They should not forget what they have studied in the 11th standard. That also they should recall, and then we can directly enter into the concept. So there are 3 sites. Students must understand the 3 sites present in the larger subunit of the ribosome. Sorry, it is not written here. I will write it with the pen, marker. A site, P site, and E site. There are 3 sites. Students should not have confusion, because by these 3 sites only the translation is going to happen. After initiation, elongation, termination. All these things are going to happen based on the role of this particular sites only. So student must understand the role of 3 sites present in the larger subunit of ribosomes, that is A site, P site, and E site. These three sites student must understand. So this is the general idea about how the A, P, and E is there. Yeah, a cartoon representation how that tRNA, charged amino acylated tRNA is coming to the A site, then it is shifting to the P site, then leaving the amino acid, the tRNA alone is exit, it is going out of the ribosome. That we the cartoon representation. Before we explain initiation, elongation, termination itself, student must understand this. Then only while we are explaining this and all, again and again we cannot explain. While we are explaining, students will easily grasp the idea how it is going to happen. So we need to say that the amino acylated tRNA, which has the activated amino acid and charged tRNA, that comes and binds to the this particular A site of the ribosome, and then it moves to the next site, that is P site, where amino acid join together to form the polypeptide chain will be formed. Then the E site, the tRNA alone leaves that from that E site. That we need to explain to the children. So quick question, teachers. Easy, very easy question.
Translation.
Translation.
Very good, easy question.
Translation.
Thank you. Easy question. You know the answer very well. To check your presence, that's all, nothing else. Now we are going to start with the initiation. Now if we explain to the children about what is the initiation, elongation, termination, for 100% sure, student will come along with us. Student will be traveling along with us, and they will have utmost clarity in the entire translation process. I think time is not up. We have time to complete. So translation. Now how it is going to happen? Initiation. The translation, first initiation, how it is going to happen. So it is a energy dependent process. So ATP energy is required, and the activated amino acid, will be present already. It is ready, and also ribosome is present. Everything is present. But somebody should start in the group discussion and all. Somebody should start the discussion. So some like that. Who is going to start that initiation? Will mRNA will start? No. Actually, it will be started by the yes, ma'am. Yes.
Initiated tRNA.
Yes, but who is going to that means especially the smaller subunit of ribosomes. The smaller subunit of the ribosome, that only will find that mRNA, presence of mRNA. Then only the larger subunit will come and bind there. Then the tRNA, everything will happen after that only. So the role played by the smaller subunit is very, very important. The smaller subunit only going to find the presence of a mRNA in the cytoplasm. Now, next quick question, teachers. Very little twisted, but easy one.
Ribosome, sir.
Identify the particular amino acid identification. Ribosome will come much later, isn't it? In the beginning itself, when the tRNA binds with a particular amino acid, the amino acylation happens. At the time, which enzyme is enzyme? Already we have seen. Yes, very good. Option D, tRNA synthetase is the enzyme. Very nice. Option D, tRNA synthetase is the enzyme. Very nice. Now, teachers, now over. Now this is bound, smaller subunits as bound to that. Now initiation factor is coming to action. The initiation factor, also called as IF, small IF, we can say initiation factor. There are totally around 9 initiation factors are there in the eukaryotic organism. In the eukaryotic organism, if you see, there are 9 different initiation factors are there. We need not to sell one by one to the children because it will elaborate the time, prolong the time, and we cannot complete the portion. But we can give a light to the children that there are different type of initiation factors are there, like ELF2, ELF2, like that 2 will be there, ELF1 will be there, like that many will be there. Same way, if you take a prokaryote, there are 3 different initiation factors are there. Though we need not to explain very elaborately to the children because it is not there in the reader, we can give an idea to the children. Student will have the clarity. That is more than enough. Now, GTP is another one, which is a very important energy currency, which is going to actually initiate the this initiation process. Otherwise, what will happen? Energy without energy, initiation will not take place. So energy must be given to that. So GTP also plays a very important role. Now, throughout this structure, GTP will be used for one amino acid. One amino acid to bind into the A site, one GTP will be utilized. Hydrolyzation of one GTP will take place for one amino acid. That will be continuously happening.
Sir, but
Now-
what is this initiation factor actually? What is the nature of this initiation factor?
Yes, ma'am. Yes, very nice. This initiation factors are the like of protein kind of things. These things only will make that binding of this charged tRNA into the subunit of this particular ribosome, smaller subunit. Otherwise, it is, see, if you take a living system, how come a subunit will know mRNA is there? It cannot know that, it cannot see it. It is a kind of chemical interaction is happening continuously between the mRNA, this ribosomes and all. When there is a mRNA is present, immediately these proteins and all will be activated, this initiation factors. They will directly go take the message and will give the information to the ribosome that there is a mRNA is readily available. Now it is your duty to come and translate. Like that, the signal will be given through this proteins only, teacher. Yes, ma'am. Yes, ma'am.
Where it will be produced, sir, actually? This
It's all in the cytoplasm, ma'am. It's all in the cytoplasm. It's readily available in the cytoplasm. Next one is the mRNA. In the mRNA, what happens? Already we know that mRNA, it will not directly start from the exactly at the extreme 5' end. The initiation will not start. There is some middle, some many sequence around even 200 base pairs also will be left. After that only the initiation will be started. Before the starting of initiation, whatever the places are there or whatever the regions are there, we call it as UTR, that is untranslated regions. That area translation will not take place. Same way at the 3' end also, after the stop codon, there are some sequence will be there. Those sequence will not be translated. They are simply persist in that we call it as again untranslated region. Both the 5' end, that is before start codon, and at the 3' end after the stop codon, in both the areas we have this untranslated region. What scientists have so far discovered is that these untranslated regions are very, very important for a efficient translation machinery. If efficient translation should take place, these are the untranslated regions are very important. Without that, it is not possible. So now we are going to the next one, initiation. Before going to initiation, I want to show you a video and then I want to come back to you. This also I want you to, can you see the animation, teachers, everybody?
Yes, sir.
Yes, please kindly share this with the children. If you want, I will also share it with you. It's available in internet also. Student must understand how the molecule level, how it is happening, not the cartoon image. Students are clear with the cartoon, but when we show the students will clearly understand how exactly protein synthesis happening. In that light green color one on the top is the larger subunit, and brown, that peach color is the one is the smaller subunit. This mRNA and this tRNA activated. The blue color one is the tRNA, and the light blue color one is actually the protein, the amino acid. The different amino acid joined together to make a protein. Can anyone say what is that band of black color is there at the top of amino acid, which I have circled? Maybe I think you are able to see that. Can anyone say, teachers?
Endoplasmic membrane.
Very nice, ma'am. Excellent, excellent, ma'am. It's an endoplasmic reticulum. Yes, it's an endoplasmic reticulum. So it's an endoplasmic reticulum. This blue color one is the activated amino acid, light blue color. Dark blue color is actually the tRNA, and the single strand of mRNA, you can see that single stretch is there in the smaller subunit. Kindly show this to the children whenever you, if it's online or in the smart board or in a, even if they're you're having communication through the WhatsApp also, make the children to understand in a living system, this is how the things are going to happen. That is where the clarity is very important.